One of the other major subdisciplines of ecology is community ecology. Obviously, species don't exist in a vacuum. They interact with other species. And whenever you have two or more species in the same area at the same time interacting with each other, you have what is known as a biological community. Now, there are several concepts in community ecology, and we're going to mention a couple here that are important for quantifying community structure. One is species richness. This is simply the number of species in a community. To use the figure here to illustrate, let's say that you have two forest communities. In this community, you see that you have 4 different species of tree. So the species richness for this community is simply 4. Okay? Well, species richness is helpful to know because the number of species has implications for how stable the community is, the number of interactions that can go on, and a number of other things. So species richness is helpful to know, but it doesn't take you very far in quantifying the complexity of the community itself. For example, let's look at community 1 again. Remember, we said that we had 4 different species, A, B, C, and D. Well, if you look at community 2, you have the same 4 species, A, B, C, and D again. But if you look closely, these two communities are not identical to each other. The way that they differ is that community 1 has a much more even distribution of those 4 species. Community 2 has a very uneven distribution. It's dominated by species A. 80% of the trees in this forest are species A. So even though they have the same species richness of 4, clearly that doesn't take you very far in understanding and quantifying the complexity that you have between the two. So that leads to another concept that is known as species diversity. This is a measure that takes into account species richness and relative abundance of each species. So it takes into account evenness, and let's just go ahead and say that high species diversity is very desirable in community ecology for reasons that we'll talk about later, some of which are pretty readily apparent. It just increases the stability of the community. So community diversity is something very important to know, and it is very important to be able to quantify it so that you can compare one community to another. Now, there are several different methods for doing this, and this is the place in the course where we're going to start to introduce some of the math. So don't let it scare you off. A lot of people, when they see formulas with Greek letters, start to get a little uncomfortable, but don't let that happen to you. This isn't rocket science. It's just math. So let me give you one example. One of these methods for calculating and quantifying diversity is known as Simpson's Index of Diversity. And the way that you calculate it is 1 minus the sum of all of the relative abundances for all of the species in that community squared. So it's the sum of all of those squares. So let's break this down just a little bit. Let's look at community 1. This is just a hypothetical community to illustrate. So community 1, it's a community of forest trees. We've got 5 species, so the species richness is 5. We've got 1,000 total trees. These are the individual population counts of these species. So one of the things that you can calculate is relative abundance, represented by the lowercase p. So the way that you calculate relative abundance is you simply divide the number of individuals of each species by the total number of species. It's really sort of like calculating percentage. You're just not multiplying by 100 to get percentage points. So relative abundance is usually represented by a decimal number here, and like I said, it can be converted into a percentage, and if it helps you to think of it in those terms, go ahead and do that. So what this chart's telling us is that about 52% of the trees in this forest are white oak, 26% sassafras, and so on. So these are all of the relative abundances. Well, to calculate Simpson's index of diversity, you square the relative abundances, and that's represented in this column, and then the formula tells us with the Greek letter sigma to sum all of these squares, and then we subtract that from 1 and we get a number. And Simpson's index of diversity ranges from 0 to 1. The closer you are to 1, the more diverse the community is. The closer you are to 0, the lower the diversity. So to illustrate, compare community 1 to community 2. We've got the same 5 species in each community, but as you can see, their relative abundances are very different. Much like the picture in the previous slide, one of these communities is dominated by one species. It's got a very high, in community 2, Virginia pine has a very high relative abundance. Over 80% of the trees in community 2 are Virginia pine. So that's not a very even distribution, and like we said, species diversity as a calculation takes into account both species richness and relative abundance or evenness. So when you crunch the numbers for both of these communities, you see that community 2 comes out with a lower Simpson's index than community 1. So this is our first glimpse of being able to take patterns that you see in nature and quantify those patterns so that they can be compared. We will work through additional examples like this in this module and also in the field when we are together for that week. One other example of a diversity index I'd like to mention, because we'll be using it in class, is called the Shannon Diversity Index, and it's just slightly more complicated than Simpson's, but it also uses relative abundance, but in this case, instead of squaring the relative abundance, you multiply the relative abundance by the natural logarithm of the relative abundance. That's represented in this column, and then you sum all of those and then take the negative of that, which is going to end up giving you a positive number. The range of Shannon's diversity index varies based on how many species there are in each community, but the thing that you need to keep in mind is that the higher the H value, higher the diversity index, the higher the diversity, and of course that makes sense. And like I said, we'll work through several examples like this as part of the homework and when we are together in class. One other metric that I'll mention very briefly that can be used to compare species richness and diversity among different communities is Sorensen's coefficient of community. And it is described as a measure of similarity of two or more communities. So there's an illustration here comparing species across 3 lakes. These numbers are just hypothetical to illustrate, but you need 3 pieces of information to calculate Sorensen's coefficient of community. One is S1, the number of species in community 1. S2, the number of species in community 2, and then C, which is the number of species that are common to both. You plug those numbers into this very simple formula, calculate it, and then your quotient here is a number, and you can simply use this to compare how similar two communities are to each other or to other communities. And we'll do you'll get an opportunity to do an example like this as well.